I'm John McGuigan. I'm a biologist at Soft Genetics. This webinar will cover rare disease studies using new features introduced in NextGENE version 2.16. This includes functional prediction scores, 1,000 genomes frequency, and advanced mutation comparison using the variant comparison tool. As the price of sequencing continues to fall, it makes sense to sequence the exomes or whole genomes of several family members in order to find mutations that fit expected inheritance patterns. Filtering for mutations that match compound heterozygous or autosomal recessive patterns of inheritance, for example, can lower the total number of potential causative mutations by a few orders of magnitude. Combining this type of analysis with functional prediction can reduce the number to a few dozen mutations, a list that is small enough to verify with Sanger sequencing. NextGENE is now able to import data from the dbNSFP database. Specifically, it imports 5 different scores and the 1,000 genomes frequency. In the dbNSFP database, all of the scores are adjusted to range from a benign or non-conserved score of 0 to a deleterious or conserved score of 1. All of the scores also have classifications, for instance, probably damaging, possibly damaging, or benign. A link to the paper that describes the dbNSFP database in more detail can be found in the NextGENE Viewer Help menu. The Convert dbNSFP Files tool in NextGENE's Tools menu includes a link to the dbNSFP website where the database can be downloaded. Right now, there are a few versions of the database. Version 1.1 is more useful because it also provides 1,000 genomes frequencies. However, the light version will also work. After downloading and unzipping the database files, they can be loaded in the conversion tool. After conversion, the original files can be deleted in order to save some disk space. When new alignments are run, they will automatically query the dbNSFP location that is listed in the NextGENE Options dialog, which can be found in the Process menu. This location is automatically set to the location that was specified in the File Conversion tool. Projects that have already been run, or projects that are aligned to human GenBank files, can be queried from within the viewer. When the variant comparison tool is used, it will load the dbNSFP information that is associated with each project, no matter if it has been queried automatically or manually. By default, dbNSFP information is not displayed in the mutation report. The information can be shown by checking the appropriate boxes on the dbNSFP subsection of the Display tab in the Mutation Report Settings dialog. This applies to individual projects and to the variant comparison tool. The dbNSFP information can be used for filtering in several different ways. The database has information for most non-synonymous SNPs, but it is possible to hide any mutations that do not have this scoring information. It is also possible to exclude those with high 1,000 genomes frequencies or with certain scoring criteria. Each functional prediction or conservation score can be filtered separately, and the mutations can be filtered on the total number of scores that are passing. In the mutation report settings, it is possible to hover on any classification in order to see the full name. For example, a polyphen 2 declassification means probably damaging. By default, all classifications are checked and the minimum score is set to 0, and so every score is passing. Unchecking benign classifications will cause functional prediction scores to fail if they were not predicted to be deleterious. If the number of passing scores is not greater than or equal to the specified number, the mutation will be hidden in the final report. Here are some settings for an example filter. The goal is to identify mutations with at least 2 scores predicting a damaging result and to remove mutations with a high frequency. First, the maximum frequency is set to 0.05 or 5%. Phylo P classifications are all unchecked so that the Phylo p-score always fails and cannot be considered one of the two passing scores. In this example, the conservation score is not being considered. Neutral or missing predictions are all unchecked, so that a score can only pass if it was predicted to be damaging or possibly damaging. Finally, the main setting is adjusted so that at least 2 predictions out of 4 functional prediction scores must pass. The functional prediction scores don't always agree with one another because they use different methods, so this type of filtering should always be used conservatively. In this example analysis, there are 6 exome sequencing projects, 2 unaffected parents, 2 affected children, and 2 unaffected children. Each project has approximately 40 to 70 million 100 base pair paired-end Illumina reads. A compound heterozygous inheritance pattern is expected. After alignment, the projects can be compared using the variant comparison tool, which is found in the Viewer's Tools menu. The Load Projects button will open a dialog. Projects are then loaded one at a time. This dialog can also be reopened from the Sample Settings option in the Settings menu after the comparison has already been run. After loading a project, several properties can be set: relationship, phenotype, and mutation type. Certain comparisons require some of this information. For instance, common heterozygous and template filtering require that the relationship and phenotype be specified for every sample. Manual filtering can also be specified by selecting a specific mutation type for each sample, such as homozygous, heterozygous, present, negative, or undetermined. The maximum number of projects can vary based on the size of the projects and the hardware of the computer the NextGENE is running on, but 10 to 12 projects are the maximum. Aside from being resource intensive, larger comparisons would risk missing out on some regions due to low coverage in any one project. The next page on the Load Projects dialog allows for one of 7 different comparison types to be selected. The first 3 comparisons only use basic information. Show All will list any mutation present in at least one project. Show Shared will list mutations present in all samples. Show Different will list mutations that differ in 2 or more samples by a minimum amount. This minimum amount can be assumed to pass if one sample has no mutant alleles by selecting the Exclude 0% mutations option. This means that a homozygous normal and a 20% mutant will still be treated as different even if the minimum difference is set to 30%. The low coverage SNP setting is useful for finding mutations that were missed by some projects due to low coverage. The mutation type comparison will use the information specified on the previous page of the dialog. Undetermined samples, either set by phenotype or by mutation type, will be ignored during filtering, but will be displayed in the report. Template filtering uses predefined or custom inheritance templates, such as homozygous recessive. Compound heterozygous filtering looks for at least 2 heterozygous mutations present in the same gene. It uses the family and phenotype information to check that one mutation came from each parent, if the parent samples were provided, and that unaffected samples do not have both mutations. This filtering is updated after other filters. This means that if score filtering or dbNSFP filtering removes one of 2 mutations found in the gene, the other mutation will also be hidden. The Check Allele Counts for Negative Mutations option will count and display allele information for negative samples instead of listing the default NA. However, this will add some processing time. Custom templates can also be created. In each template, there is information to convert family and phenotype information to the correct mutation types. For example, in an X-linked recessive comparison, we see that both parents are unaffected. This means that affected sons must be homozygous and affected daughters should not exist. When the comparison is first opened, the mutation report is visible. It is the same as a normal mutation report, except information for each sample is laid out side by side. One button on the toolbar will allow the selection of projects to be displayed above the mutation report. The alignments are kept in sync and are displayed side by side in the same order as the samples in the mutation report. If a compound heterozygous comparison is made, another button will show a report listing all possible pairs of mutations. Alternate shading indicates different genes. In the projects used in this analysis, a mutation was called if there were at least 3 occurrences of the SNP allele, or 20% of total coverage. These 6 projects had over 350,000 mutations between them. Approximately 41,000 were possibly significant, meaning that they were non-synonymous SNPs, consensus splice site mutations, or INDEL's encoding sequence. When using common heterozygous filtering, the number was reduced to 669 mutations. 91 of these were potentially significant. Approximately 70% of these had dbNSFP information, and they were filtered out if they did not have at least one conserved or deleterious prediction, or if they had a 1,000 genomes frequency greater than 5%. This filtering left 45 mutations. After some confidence score filtering, there were only 12 mutations left, which made up 7 possible pairs of mutations in 5 separate genes. One of the final 7 pairs of mutations was in the 4th coding sequence of the KC-TD7 gene, which is known to be involved in the disorder that was being studied. Both mutations were classified as conserved, and one was classified by polyphen 2 as damaging. We would like to thank our collaborators in the Undiagnosed Diseases Program at the National Institutes of Health for providing the sample data and collaborating in the design of this tool, including Drs. William A. Gall, David Adams, Thomas Markello, Murat Sinkan, and Cornelius Borgel. To recap, 2 new tools have been added for NextGENE 2.16 in order to assist with the study of rare diseases. The dbNSFP database can be imported to provide functional annotation, and the variant comparison tool now allows for filtering mutations by several different inheritance patterns. Using these tools, it is possible to narrow down the list of potentially causative mutations from several thousand to several dozen in only a few minutes. Thank you for listening to this webinar on the use of NextGENE for rare disease studies. For more information, please visit our website at softgenetics.com or email us at info@softgenetics.com.